Hey, welcome back. Today I'm going to be talking about the Hardy-Weinberg equilibrium and then the violation of such with selection. I'll be using a made-up parrot species that comes in three colors and expresses them through incomplete dominance. Blue is homozygous dominant, purple is the heterozygote, and red is the homozygous recessive. And I will be using R as my allele. So first off, we need to look at the amount of individuals, then take those numbers and put them through equations to find the genotypic and allele frequencies for the Hardy-Weinberg assumptions. For allele frequencies, we're going to take 2 times the homozygote, add the heterozygote, then divide by 2 times the total. For big R, we get 0.59, and for little r, we have 0.41. Well, now we can calculate our genotypic frequencies. Our big R and our little r are equivalent to p and q respectively, so we can easily plug in our numbers to get the different parts of the equation. We find that blue parrots have a frequency of 0.34, purple parrots have a frequency of 0.484, and red parrots have a frequency of 0.168. This is all under assumptions of Hardy-Weinberg, but what happens if we violate the equilibrium with selection? Well, we have to look at how that affects the equilibrium and the frequencies. For this, remember that our initial genotypic frequencies were big R equals 0.35, big R equals 0.48, and little r equals 0.17. Now I have given some fitness amounts for each genotype. Big R sits at 0.5, little r sits at 0.7, and then big R is at 1. For calculating our unadjusted survivors, we just multiply our initial frequency by that genotype's fitness level. You can see our values in the chart. To find our average fitness, aka W, we just add up all of our unadjusted survivors, and in this case we get 0.774. Finally, to get our adjusted frequencies that show off selection, we take our unadjusted survivors and divide by our average fitness. So for red parrots, we take 0.119 and divide it by 0.774 to get our new frequency of 0.154. We get 0.62 for purple parrots and 0.226 for blue parrots. Now we find the new allele frequencies. So to find the frequency of R, we take 0.226 plus 1/2 of the heterozygote, and that equals 0.536. And then to get the frequency of little r, we just do 1 minus that, and that gets us 0.464. And when comparing initial and post-selection frequencies of alleles and genotypes, we can clearly see heterozygote advantage showing itself as a stabilizing selection. We also see a change in our alleles with big R going down and little r going up. Perhaps the purple heterozygotes have gained selective advantage over others due to camouflage. In this hypothetical tropical forest, many plants have a light purplish hue that the purple parrots tend to blend into better. These plants tend to be in shady understories, so perhaps the bird's favorite food has decided to move down into the shadier regions of the forest. If this frequency trend keeps going, red and blue parrots will become the minority, while purple parrots will be much more common in comparison. So thank you. That is it for Hardy-Weinberg selection. I look forward to seeing you in the next video. Bye-bye.